on this computer. All right, we are recording. So I'm going to share my iPad so that you guys can see this. And remember, if you want to get those 2 learning targets and you've maybe fallen behind a little bit or a little bit unsure, then this is definitely the day for you. Even if you haven't and you're right on pace, this is another way to think about, oh yeah, this is how to do it. And then if one of your friends wants to do this and hasn't made it to the seminar, then you could teach them how to do it and you'd be a hero. All right, so I'm gonna start here with my little Canvas thing. I'm gonna click on Canvas. All right, I'm gonna click on our EA Science, what have you here. Oh, and for some of you on here, I put in a whole new module called Seminar Tech Tips, and it shows you how to get an assignment from Canvas to Notability, how to use Notability, how to put Notability back to files, and then submitting something to Canvas. We don't have to submit anything for EA into Canvas for this one, but some of your other classes, you might have to do it, and that could be a helpful video if you don't know how to do it. All right, so first things first, let's grab our formative project right here, DNA formative project, and I'm gonna click on this nucleotide to gene project. And here is the thing that you all recognize that you've been working on right here. And now I'm going to send this off. I'm gonna send it over. And I'm not gonna go to Notability right away. I'm gonna go to copy to Notability. And I'm gonna create a new note in Notability. I'm gonna import this. Here it is right here. I'm going to see if I can make this demo and then the date today, 10 November 2020. So we know that's our demo for today. All right, so we're good with this demo right here. The next thing that would have happened is heading back over into Canvas here. The next thing that would have happened, back it up again, is you would have been assigned a nucleotide sequence that you had to uncover, you had to unlock. And that came from this student formative nucleotide list. And so I'm going to click on this list and it opens up this doc here, and you'll recognize some of your names there, which ones you've been assigned. For this one, I'll just grab HS19 here. All right, so I'm going to click on HS19. And I have my structure is set right here. And I have to figure out the code of this structure. All right, so I'm going to see, woo, come back. Oops. Oh, it's just going forever. I'm going to see if I can pull Notability over here. All right, woo. Bring this one back down here. And to make sure you're not all bored here, I am going to quickly, as I can, write in this sequence. So I'm going to go in 3s here. Let's see. And I see that first one is A. The second one is G. Third, that's 3. That looks like C to me. And I'll go another 3 over. 4, that is T. 5, that is G. 6, we got G again. And let's go the next 3 codons. So 7, 8, and 9. I see that as being C. That one there, number 8 is T. Number 9 is also T. And let's go 10, 11, and 12. That is, I'm trying to go as fast as I can here so that you don't get too bored. That is C. that is T. Number 12 is G. And 13 here is T. 14 is also T. 15 is C. All right. And we have 16, 17, and 18 here. 16, I can see that that is C. 17, I can see that is T. 18 is G. Now we got 19, 20, and 21, our final three here. That is G. That is C. And finally, that one there is C. All right, whew, that was quick. So now we don't need that anymore. And we have notability here. And now I just need to write the complementary strand. So maybe I'll just make that a slightly different blue. So the complement to this is T, C, G, A, C, C, G, A, G, A, C, A, G, G, A, C, C, G, G. All right. And now my next part 2 we learned is I need to transcribe that original sequence into RNA. So I showed you the trick on how to do this before is you basically just copy this one up here. So I go A, G, C, U, because there's no T in RNA, G, G, C, U, U, did I do that right? Yep. C, U, G, U, U, C, C, U, G, G, C, C. All right. So without having to do much else, I wish I would have put a codon wheel here, but now I need to look at a codon wheel so that I can translate these 3-letter codons here. So maybe I'll block these out like this to show you this. And if I don't have a codon wheel, I'm just going to pull up here. I'm going to, oops, come on, grab Safari over here. I'm going to add a tab. I'm going to say codon wheel. I've done this a million times. I find that codon wheel that I like. Oh, Securely's helping us out here. Let's go to images for that codon wheel. Ooh, I like this one a lot. I'm just going to take that and, whoops, and bring it on over into Notability here. I don't need that Safari anymore. And then I can bring this down here. And my first codon is AGC. So AGC is serine. So that's serine. And my next codon is UGG. So UGG is tryptophan, TRY. My next codon is CUU. CUU is leucine. My next codon is CUG. CUG is leucine again. And here we go. This codon, the next one is UUC. So UUC is phenylalanine. So PHE. Getting close to the end here. CUG. CUG is leucine again. Ooh, that's some nice handwriting. Leucine again, and finally ending with GCC. GCC is alanine. Alanine. All right. So we've made it up to the beginning of last week with what we learned. And your teacher will give you a specific mutation at one of the 21 nucleotide positions. And now I have to say if my amino acid code is going to change. So where is my mutation? I'm supposed to get a mutation from my teacher. Well, if we go back into Canvas here, I was doing HS19 right here, and it says at position 19, the original nucleotide was G, and now I need it to be A. So position 19, oh, that makes sense because HS19. Position 19, G switches to A. So position 19, G switches to A. So at position 19, so this is 21, 20, oops, sorry, I'll show you. 21, 20, 19. Right here, this becomes A, which means this would become T, which means this down here becomes A. So this would mean that all of this would stay the same. All of that would stay the same, but perhaps that nucleotide might change. So we talked about a cool way to do this might be to get my scissors up here, my scissor tool, and kind of lasso all the ones that would stay the same. Copy that. Paste it down here. And, oops. Fill it right in there so it looks all nice and pretty. And then we could even, if we wanted to, I suppose we could crop this a little bit so it looks even better. Sure. All right, that looks pretty good. And now we see that this ACC probably doesn't code for alanine anymore. So what is that going to be? Well, ACC codes for threonine instead. So this becomes threonine. So we did have a mutation in our protein sequence. Amino acid number 7 changed from, what was it originally? Alanine to threonine. And there we go. So I am going to